Respected dignitaries and members of the audience, a very good evening to all of you. I, Raksha J. Nayak, M.Pharm student from the Department of Pharmacology, MCOPS, welcome you all to the inaugural lecture of the Diamond Jubilee Commemorative Lecture Series. We feel immensely delighted and honored to have with us today Dr. M.V.S. Valiathan as our chief guest for the evening. A hearty welcome to you, sir. Professor Marthandavarma Shankaran Valiathan graduated in medicine from the Kerala University and received training in surgery in the UK with subsequent specialization in cardiac surgery in the US. He became a fellow of the Royal College of Surgeons of England in 1960 and a Master of Surgery from the University of Liverpool 2 years later. He received the fellowship of the Canadian Royal College in Cardiac Surgery in 1970. Professor Valiathan served on the faculties of the Georgetown University Hospital, Washington, Postgraduate Medical Institute, Chandigarh, and IIT Chennai, before moving to Thiruvananthapuram as the architect of the Sri Chitra Tirunal Institute, Trivandrum, where he remained as a professor of cardiac surgery and director for 20 years. Subsequently, Professor Valiathan served as the first Vice-Chancellor of Manipal Academy of Higher Education. He is a former president of the Indian National Science Academy, Association of Indian Universities, and currently a national research professor. Professor Valiathan's professional and scientific interests were cardiac surgery in children, studies on the causation of a tropical heart muscle disease, and the development of a technology for cardiovascular devices such as the Chitra TTK heart valve. Dr. Valiathan changed his field of interest to the traditional medicine of India in 2000 and published redactions in English of the 3 authoritative texts of Ayurveda, Legacy of Charaka, Legacy of Sushruta, and Legacy of Vagbhata from 2003 to 2009. He has organized a task force in Ayurvedic Biology with the support of the Department of Science and Technology, Government of India, which funds research projects in modern biology based on Ayurvedic cues. He is a recipient of the Fellowship of the National Academies of Medicine, Science and Engineering in India, many honorary doctorates, awards and prizes in India, and several international honors including the Hunterian Professorship of the Royal College of Surgeons of England, Chevalier in the Order of Palms Academiques of the French government, and Dr. Samuel P. Asper Award for International Medical Education, Johns Hopkins University in the US. He received the Padma Vibhushan from the Government of India in 2005. Sir, it is indeed an honor to have you with us today. I now invite our chief guest, Dr. Valiathan, onto the dais to deliver the inaugural lecture. The topic of today's lecture is Ayurvedic Biology. I request all the members of the audience to kindly restrict your movement and keep your mobile phones on silent. Over to you, sir.
And it is wonderful to be here today, especially celebrating the Diamond Jubilee. And you know, this was one of the institutions established by Dr. T.M.A. Pai. It started as a school, I think, initially, giving only a diploma. Subsequently, it has grown. It is one of the, I think, the top-ranking colleges of pharmacy throughout, I think. Now it has postgraduate courses in every subject. So it is one of the top institutions of MAI. When I was asked to come here and speak, I gladly accepted. And the subject I chose was something which could be of interest to you, and that is Ayurvedic biology. Ayurvedic biology, that term first appeared as the title of a decadal vision document published by the Indian Academy of Sciences, Bangalore. As some of you will know, this was the academy founded by Sir C.V. Raman. It's one of the top science academies of the country, and they published this in 2006 towards Ayurvedic biology. That was the first time that term appeared. Now, what is Ayurvedic biology? Ayurveda is Indian. Biology is European. as we understand it. So Ayurveda is east, biology is west. So this encounter between east and west, it started long ago. I would say more precisely 16th century. There was a doctor by name Garcia de Horta. He was a Portuguese physician. And they had just conquered Goa or occupied Goa. And this Garcia, he came and he set a practice in Goa. And he practiced medicine there for 36 years. And not only he practiced medicine, he was a keen observer. And he recorded what he saw, what he experienced. And he published a book called Colloquies on the Simples and Drugs of India. That book described diseases. It described treatment followed by the physicians in Goa at that time. Not only Ayurveda, there were many other practices, local practices. He describes all of them. And he also described for the first time Ravolfia serpentina, which is not mentioned in Ayurvedic texts. Charaka Sushruta Vagpata. You will not find Ravolfia serpentina. But this was being used there. So many interesting things and very vivid description of diseases. Like cholera. In Ayurveda we have Atisara. It is described, but just 2 lines. But in Western practice they have description of diseases. It has been made an art. Like somebody with angina pectoris, Parkinson's disease, how the patient walks. These are really painting in words, beautifully done. You read it for the sheer pleasure of it. Now Garcia has done some of that. For example, talking about cholera, which we, 2 lines we dismiss. Here there is a detailed description how that cholera patient suffers, symptoms, signs, the suffering, all that. So it's a very interesting book. That book is written in the form of a dialogue. He is a partner in the dialogue, and the person who has the dialogue with him, there are 2 different subjects. One person, the Garcia himself, when he says I, he is a conservative European physician. But that student, the partner in the dialogue, he is coming up with new ideas. We are seeing some of these things being done by the natives here. Can we try that? So he says, no, you cannot try that. So like that is interesting. So the dialogue form of presentation was very popular at that time. Now that book set off a great deal of interest in medicinal plants of India. This was translated into many European languages, became very popular. For the first time, the world recognized that there are medicinal plants in India. And they were interested because many of the European countries, like France, they were all trying to colonize. They wanted to come to India because India had a lot of wealth. So they all wanted to come here. And they must know something about the illness here, because tropical diseases they did not know. Snake bites, for example. They have no idea how to treat. So they have interest in this book. That's how it became very popular. Now that process, once the Europeans got interested, plant studies, that became extremely popular. Taxonomy, what we call. So taxonomy was for almost, from then it started, 17th century, there was a Kochi, there was a Dutch governor called Van Reed. He was from Holland. He got interested in medicinal plants. And the reason he got interested, he found the Malayalis, he was living in Kochi. He thought the Malayalis, the diet was hopeless, very bad, nutritionally, very poor. But the people were very hardworking, very productive. How could these people eating this miserable food, how can they be working so hard? There must be some secret. And he thought in all of the food that we cook in Kerala, we always have coriander, pepper, turmeric, some of these spices. Without that, we cannot cook our meals. So Vandry thought maybe this is what is giving them the strength. So he wanted to explore this, and he organized a team of almost 100 people, herbalists, Ayurvedic. There was one Ayurvedic physician to tell them, because the Ayurvedic physicians were all high caste people. They wouldn't cooperate with foreigners. So they found a low caste Ayurvedic physician who was a really great physician, Itti Achyutan. He was drafted in this. Excellent plant drawings, that Hortus Malabaricus, 30 years of effort by this big team, that was published in 10 volumes in Amsterdam. That was the next phase, again taxonomy. This continued up to almost 20th century. Even now taxonomy continues, the interest in taxonomy. But 20th century something new happened. I'm talking about the East-West encounter, Western science encounter with Ayurveda. 20th century, all of you maybe heard about Ramnath Chopra, the father of Indian pharmacology. He had done a PhD from London. He came back. He was appointed as a professor in the School of Tropical Medicine, Calcutta. Ramnath Chopra was undoubtedly the person who started preparing compounds from medicinal plants. He went one step further, studying isolating compounds, purifying them, studying the effect in vitro and in vivo, toxic effects of plants. All these, and extensive studies were done. Number of people worked under him, and he was known as the father of Indian pharmacology. And in his writings you will find he always wanted to develop drugs from these medicinal plants which can be used by all systems of medicine, including modern medicine. That was his aim. So that started the change from taxonomy. The approach changed to pharmacology, looking at medicinal plants. And this soon led to natural products chemistry. And we had people like, that was almost mid of 20th century, Govindachari, Asima Chatterjee, all these people doing extensive work on natural products chemistry, extensive literature, thousands of papers. And even today, that database is used by even multinational companies. They are searching for drugs for degenerative diseases. They look at this database. Information is there, chemical composition and so on. So India became something of a leader in natural products chemistry. But biology did not stop with that. 20th century, as you know, the greatest discovery was double helix, which not only revolutionized biology and medicine, but there is no field of science which has not been touched by double helix. Now, naturally, that window has never been used in studying Ayurveda. We only stopped with the natural products chemistry. Now, this is where a new beginning, because we are looking at through a different window, Ayurveda. And when you apply the techniques of molecular biology, or modern biology more correctly, then you begin to see a different type of Ayurveda. And that is Ayurvedic biology. You are actually doing biology as rigorous as any other branch of biology, but the cue for this always comes from Ayurveda. That's the only difference. So in fact, the whole thing started, I gave a lecture on this in 2005 in Banaras University, and Professor T.V. Ramakrishnan, who was the president of the Indian Academy, he was chairing the meeting. So after I had finished, he came and asked me, can you please write this? Because in the academy, we published this decadal vision document. I would like to publish this. So please, so I wrote that. And I gave specific example, what the history of the background of Ayurveda, what this Ayurvedic biology is, what does it mean, are there specific examples, instead of saying cues from Ayurveda. give us some cues. So there are 5 cues I have mentioned. Now this is difficult because when you read an ancient text, Charaka, Sushruta, Vagpada, Charaka was 1st century. Sushruta was earlier, but the version we have is 4th century. Vagpada is 6th century. So these are all written many hundreds of years ago. And they are often very terse, what they write. Almost like a sutra, maybe 2-3 lines. They don't give any details. Often they don't give any explanation. Now from this, how can you develop something which can be made into a project? Science means experiments, not mathematics. I'm talking about our biological sciences. So if you want to do experiment, you need to write a protocol. So is it something that can be converted into a project? Identifying that, that's the first problem. Having identified the area, you have to write a specific question, write a protocol. Are you going to do it in animal? Which animal? All the getting into more and more details. That is very difficult. You can't get any helpful information from the old texts. You have to talk to traditional schools of Ayurveda. They may be practicing this. After all, Vedas, 1,000 years simply by oral tradition they maintained it. So we should not underestimate that tradition. If there is a traditional school which has been practicing Ayurveda for hundreds of years, that information is important. You should not dismiss. With all this help, you can write a protocol. That is necessary. Then you have to find, to do a project, you have to find an Ayurvedic physician who is scholarly, who is inquisitive. You have to find such a person. It's very hard to find. You may find any number of Ayurvedic physicians, but they won't be scholarly. They may not be interested in any research. That won't do. And we need a very good scientist who is open-minded, who is willing to accept that there is something to learn from Ayurveda. You have to find such a compatible team. And then, above all this, you need money. To do these projects, large sums are required. Who is going to give the funds? Because this will not find a place in the thrust areas of any department. You go to science and technology, you go to health ministry, whatever ministry you go, even Ayush ministry. At that time there was no Ayush ministry. But nobody will give money. So these are the formidable obstacles. And the only person who really helped, I went around after these 3 books were published. I wanted to know how did the scientific community look at this whole idea. So I went to several science institutions, very big institutes like Indian Institute of Science, Tata Institute of Fundamental Research, National Institute of Immunology. People I knew, they were inviting me to go and speak. What I found was there is very great interest. My subject was the call of Ayurveda for the scientist. That was my subject. I simply wanted to know how much interest they had in this. Very great interest. TIFR, I remember, is a fundamental science, physics and mathematics. There were 45 minutes, there were questions. So much of keenness to know. So there is great interest, no question about that. But when it comes to solving these, I had difficulty. Until Dr. Chidambaram, who was a neutron physicist, chairman of the Atomic Energy Commission, he was the one who gave the support. He had read this towards Ayurvedic Biology in 2006. He told me, I have read this. You have mentioned half a dozen things which can be done. We are not a grant-giving agency, but I think these are important things. I'll give you support in the beginning. If that works, somebody will take it over. That's how it all started. I won't get into all these. The subjects which I suggested, some are concepts, some are procedures, some are products. The concept, the best example is that of Dosha prakriti. Ayurveda has this concept of Dosha prakriti, Vata, Pitta, Kapha. Now, these are constitutional types. A great deal of injustice has been done by thinking Vata is wind, Pitta is bile, and Kapha is phlegm. I myself used to say this. How can you have a system of medicine based on Vata, Pitta, Kapha? Does it make any sense? I used to say this. But that's not what it says. Vata, Pitta, Kapha are constitutional types. That is, certain physical characteristics, mental characteristics, behavioral characteristics. When they combine in a certain proportion, that individual has that constitution. Vata, Prakriti, everybody has it. Everyone has a Prakriti, not in the same proportion. And that Prakriti is determined at the time of conception. It cannot change. This is very fundamental. And in Ayurveda it is important because that tells you the predisposition to diseases. Vata, Prakriti, they are prone to get certain diseases. Pitta, certain. Now this is fundamental. Predisposition to diseases. Second, the response to treatment. That also differs. The same treatment will not work the same way in all the three. So we are coming somewhat to personalized medicine. You have to look at the person, but then you can treat his constitutional type. Now these two very, very important considerations for Prakriti. So determining the Prakriti is the very first thing that you have to do when you give Ayurvedic treatment. But is there a biological basis for this? Traditionally, a physician would sit down with the patient, talk to him, observe him. There is no laboratory tests or anything. But in our experience, we have done about 45 minutes a traditional physician would spend. Like my Acharya Raghavan Tiruvilpad, he would spend about 45 minutes on an average. You cannot have an OPD with 200 patients. You cannot do determination of dosha prakriti like that. Now there they observe, from those observations, physical, mental, behavioral characteristics or traits as they are called, they say this is this prakriti. That's how it is determined. Now that it will be difficult because inter-observer variations are there. Today it will be difficult for us to convince a scientific community. Based on this you are going to do molecular studies. If you, same patient goes to another person, another Vaidya, he may find something else. Or the same physician, after a month, he may change something else. How do you know that won't happen? So these were all the issues in determining the Prakriti. Is there a biological basis? Ayurveda talks about these three constitutional types. In that regard, can we use Prakriti determination in these? If there are three different types, Vata Prakriti people, Pitta Prakriti, Kapha Prakriti, then we are making a major advance, a classification of humanity based on doshas and their proneness to diseases. All that we begin to understand. But what is the biological basis? Now that is the first project, a concept trying to do that. Second was rasayanas. Rasayanas are, I won't get into details, all the five we have mentioned very clearly like this. That is a procedure rasayana. There is also a conceptual basis there. And now that we can, when rasayana is given, it is toning up the whole body rasayana. What happens in the body? Is metabolism, various other things we can measure. Because all along we have been looking at only the phytopharmacology. We have never looked at the body's response, physiology. What is the change that's happening in the body physiology, body response? What is the immune response? These are all very important, but we have never looked at it at all. So with this Ayurvedic biology, we have completely changed. Not that phytopharmacology is abandoned. That will continue. Search for drugs. That is separate. Here we are looking at biology. Body's response in very many ways. So this was the, Rasayana was the second. And we chose Amalaki Rasayana, which is one of the commonest used. which described in every Ayurvedic text. That is the Rasayana. We chose, extensive studies were done, including in Manipal. And the third was Panchakarma. Panchakarma is used as very, very commonly, as you know. That is part of the treatment, along with drugs and so on. But when Panchakarma is done, what is the body's response again? For example, obesity is being treated by Panchakarma. Basically, it is enema which is being given. But what changes do they produce in the system? In fact, first time this idea was being considered, we wanted the Tata Actrac in Bombay, which is the Cancer Research Institute's biggest laboratory. And Dr. Chiplunker, the director, she's a very well-known immunologist. I requested her for help in doing these immune studies during Panchakarma. Panchakarma was being done by Podar Institute, which is a very well-known Ayurvedic institute of the Maharashtra government. She said, you know, Dr. Valiathan, now this is enema being given. That's not correct. Panchakarma is not enema. But she told me, you know, this is enema being given. Do you really think it will produce any changes in the immune system? This is what she told me. So I said, first of all, it is not enema alone. These patients undergo a certain pre-treatment. There are many things, preparations are done before the enema is given. And secondly, weight loss is there. And with that, is it conceivable some changes in the immune functions? Can that be measured? So with some reluctance she agreed. But I must say that after the work started, a few months later she called me. She said, Dr. Valiathan, I'm surprised. These are insulin resistance promoting cytokines. Obesity, for which this was being given. Obesity is actually not adipose tissue sitting here and there, big lumps of it. That was our concept when we were students. But today we realize these are actually like endocrine glands. They are producing, they're very active. And one of the things is these insulin resistance promoting cytokines, IL-6 and so on, a whole series of them. So in these obesity patients who are being admitted for treatment in Podar, their levels are very high. They have an inflammatory disease. But after the enema started, 2 days later, 48 hours later, this is coming down. So she called me and said, you know, I'm surprised how this is happening. That paper is published. I am quoting only from published work. So that was the Panchakarma was another project. Then we had the old question of Rasa Sindur. We have in Ayurveda, not often used, but it is used. We call it Bhagarathvajam in Kerala. But it is used all over, mercury bhasma. And mercury, as you know, we will not touch it. It is a banned. Even dental amalgam, we will not use mercury. Highly toxic. But if you talk to Ayurvedic physicians, they use it. Siddha, they use much more. I asked P.K. Warrior, venerated person in Kottakkal. He said, we don't use it very often, but we have not seen these renal shutdown and problems like that. This is what he told me. Now that Rasa Sindur, there is a puzzle here. It's no use saying this is nonsense. That is not science. You have a highly respected physician like P.K. Warrior says, I have used it, I have not seen. And in Ayurveda, this is a source of knowledge. Today, you know, in logic, pratyaksha, what our senses can appreciate, that alone is taken for. And inference, from what we infer from that. But in Ayurveda, Shabda is also there. That is the word of the Apta, a teacher, like P.K. Warrior. 50 years he has been practicing Ayurveda. A man incapable of telling untruth. Man of great character, great humanity. Such a man's testimony is also a source of knowledge. At least I believe. So when he says he has not seen this, there is a puzzle for us. Now this bhasma, mercury bhasma, we asked the Tata Institute of the BRC. They have excellent analytical laboratories, very modern equipment. They did the analysis for us, microstructure of Rasasindur. And what they found was there is a nano transformation taking place. Very much like nano preparation which they make in the lab. They have compared, that paper is published in Journal of Synchrotron Radiation. These are all available, which received a lot of attention. So nanocarbon, for example, nano form of a substance, its properties have very little in common with the bulk. So nano mercury is very different from bulk mercury. That could be the basis for the non-toxicity. This was another project. And the last project was, which was done by CMAP in Lucknow. And that was, there is a place in Sushruta says that there are plants which are specifically effective against Vata, Pitta, Kapha. That's so I don't see that in Charaka anywhere. But there he specifically classifies. In that case, is there something like a Vata, Pitta, Kapha type? In plants also. In plant molecular biology, can we look for something like this? We were told because plant genome is very unstable, much more difficult to work with it. But there are apparently techniques like CISA, which were supposed to be very effective in doing this. This work was done by Lucknow, CMAP, published also in some international journal. Now these were the 5 areas that we identified in that document in 2006. All these have been done. So I simply want to give you 1 or 2 examples. That is, take the dosha prakriti for a start, the very fundamental concept. It was done by several institutions put together. Manipal was one. Our SDM college in Udupi was a partner, both these from here. And Bangalore we had FRLHT, Vaidya Gangadharan was associated at that time, Darshan Shankar. So that group, they were the participating from Indian Institute of Science, Kondaya. So 2 institutions from Bangalore. From Pune, the Pune University, and Vaidya Nanal, a very well-known Ayurvedic physician. And Hyderabad, the CCMB, outstanding place in molecular biology. So these were all the institutions. Samples were collected from Udupi, from Pune and Bangalore. So we had 3,400 subjects, healthy subjects, they are not sick. And the samples, once they collect, the selection is important. There are Ayurvedic people. In all these, there are Ayurvedic and modern science. That is how they are partners. So when they look at it, these various traits which they observe, they put it together. And if the Vata, for example, let us say they talk fast, they walk fast, they eat fast, they are very quick in doing everything. That is one feature. Another type may be very slow. They are slow speaking, slow in everything. One type may be, they see blood, they will faint. Another person is totally unaffected. All these are there. But the Ayurveda texts don't say the score. One is more important than the other. How do you differ? That is not there. These are all mentioned, the traits. So our method, what we followed was, these 3,400 in these 3 places, they are seen by an Ayurvedic physician. And he grades it according to their traditional criteria. But we wanted to make it a little more rigorous. And the way we did it was, there was a group in Pune in 2011, one Mahali, I have never met that person. But they had a 5-year exercise of University of Pune, CSIR, and 1 or 2 other institutions. This group worked for 3 or 4 years, I think, and did this. And what they did was, they took all these traits which are mentioned clearly in the traditional texts, and each trait they have given a score. Ayurvedic physicians know A is more important than B. So they will give score. So we have all these traits now, they can be converted into numbers. Based on that, they develop what is called IUsoft. So it is a computer-aided determination. But it had never been used. They published, but it remained there. We found this paper, we consulted them, and then we realized they found it interesting. Out of sheer interest, they did it, published it, and forgot. But what we did was, traditional determination is done, then the same subject goes to a young Ayurvedic physician with MD in Ayurveda. He had gone to Pune and learned from them how to do this evaluation using the computer. It takes only 2 weeks. And it is blinded. This young person doesn't know what the traditional determination is. And where these two, I was actually afraid there may be no correlation at all. I was worried. But when this was done, and there was Dr. Nair, who was the professor of statistics here, he was in charge of this blinding. So at the end of this, he told us there is an 80% agreement, which is very good, much more than, we have published that as a paper, this comparative study. Now those subjects, out of 3,400 subjects, we had to select, and when the traditional method, there are 3 prakritis. These are not equally balanced. There is always a dominance of one, usually. So Vata, Pitta, Kapha, all 3 are present in all of us. It's not only one prakriti or 2. All 3 are present, but one will be dominant. So when we say Vata Prakriti, he has all the 3 prakritis, but Vata is dominant. All those traits, they will be dominating. So selecting out of 3,400, we want to select only those with marked preponderance of one dosha, because that is where we are more likely to see changes, distinctions at the molecular level. If it is a mixture, it becomes very difficult to do this. So therefore, we arbitrarily decided 60% if a person is kapha, then we'll take him. 60% is pitta, we'll take him. 60% vata, 60 we use an arbitrary cutoff. That is a dominant prakriti of that person. Out of this 3,400, only 262 we could get fulfilling this requirement. That is dominant of one dosha. Then the question of molecular markers. We want to look at the molecular level. We selected single nuclear SNPs. Very commonly done, all of you will be knowing this. So we selected 1 billion SNPs. And the method was very extensive statistical analysis. Almost 1.5 years, I think, we took on that. What we are trying to do is exclude all the geographical affiliations, linguistic affiliations, geographical affiliations, all those are excluded. And finally, you have only the genetic markers. That only 52. That's all we could get. But in the referees, when we publish this in Nature reports, the referees are extremely critical. They look at very, very smallest things they look at. But even they agreed, this 52 is enough for us to differentiate these 3 prakritis, 3 phenotypes that we are looking for. This number is enough. So principal component analysis was done, and all these 3, there is accumulation in these 3 phenotypes, Vata, Pitta, Kapha. And that paper was published. And not only that, we found one gene, one PGMI, the gene, which was always associated with Pitta. And this is at the center of many metabolic pathways. And that is interesting because Pitta is tapati, heating, cooking, transforming. That is Pitta. That is metabolism. So there is it looks like there is a genomic basis. That is the claim of that paper. I won't get into the details, but that tells you essentially what this is all about. But the other projects like Rasayana, I had a good friend, Dr. Subbarao in Hyderabad. He had been working on aging for many years. And his method of study was the DNA of neurons and astrocytes in the rat brain. He would study at the 3 months, 9 months, 15 months. This was his standard method for several years, 20 years he had been working. Number of papers, he has his own assay system and all that. So I asked him to, Subbarao, can you, in this, your rats, suppose you give them rasayana and see what happens. He said, that's no problem. I don't know whether they will take the rasayana. So that was the reaction. But he did that. That paper, you will find in these 3, 9, 15 months, the DNA chain breaks, single chain, double chain breaks, they will keep on increasing. It is a linear progression. 2 years only they live. So that is, number of papers are there. But when he gives this pre-treatment with rasayana, amalaki rasayana, you can see, even a non-scientist like me, I can see that there is no such a linear. It is almost flat. So that the genomic stability is much better preserved with these rasayanas. And this paper was seen by Dr. Lakhotia, who is, some of you may know, he is the authority, one of the authorities in Drosophila research in India. So he called me and asked, do you think I can give this for my flies? Again, the question is, we don't know whether flies will take this rasayana. But when he tried it, that first paper which they published in PLOS 1, he says they take it and their average lifespan is increased. And biomarkers like starvation tolerance, thermal tolerance, these are all increased. Fertility, number of eggs laid, they are increased. All these are there. And genetically modified Drosophila for Huntington's chorea and Parkinson's, these expressions are diminished or muted even. So these are all published work. I am simply quoting the results. So therefore, a number of important findings have come out of this new knowledge. And what will happen in the future for this type of research, I cannot predict because how useful is this science? That is really not a legitimate question in doing science. We don't do science for that's a different thing, technology development. But science is new knowledge. That's what we are looking for. It may or may not be useful. May be useful much later. That is, I can't answer that. But what has happened in Ayurveda historically, Ayurveda has not been static. There is progress, very, very slow. For example, today, we all know that Nadi Shastra, pulse. There are many Ayurvedic physicians who treat using Nadi Shastra. And it is accepted as part of Ayurveda. But that is not part of traditional Ayurveda. Charaga Sushruta Vagpada, there is no mention of pulse. It came from somewhere else. We don't know whether it is Chinese. We don't know where it came from. But that is today accepted. It has become part of Ayurveda. Ayurveda has been enriched by new knowledge. Or take, for example, mercury, Rasa Shastra. That was not part of Ayurveda. In Charaga Sushruta Vagpada, there is no mercury. But today, it is a very large section. Subject in Ayurvedic colleges. Examinations are held. It's a very important subject. That is also accepted today. So similarly, tomorrow, new ideas coming. Evidence-based Ayurveda, for example. Everybody wants evidence-based. We were talking about it just now. If tomorrow there is an idea of evidence-based practice in Ayurveda, you develop a method which will be internationally acceptable. Evidence-based is not just saying something. You have to do a trial, a clinical trial, that a particular drug prepared in a certain manner should be constant, given for a particular disease, that diagnosis must be confirmed. Such a patient is treated with this and this alone. Ayurvedic drugs, entirely Ayurvedic protocol. Whatever they say, Panchakarma, it can be added, because WHO, as you know, has liberalized the criteria for clinical trials for traditional medicines. You don't have to do double-blind randomized control trials. patient is his own control. That's one. Secondly, a treatment in Ayurveda is not one alone, one Ayurvedic medicine being given. There is always diet restrictions are there. Fasting is there. Panchakarma is being given. Number of things are combined. But in modern medicine, that cannot be used for clinical trial and so on, because we have to individualize them. What does each one do? WHO says that it's not necessary for traditional medicines. It is treated as a black box. All this exactly, but you cannot change from every case you are doing something different. Then it cannot be done. But an Ayurvedic protocol, all that they traditionally do, that can be done. That can be, clinical trial can be done. That is evidence. So if that concept of clinical trial as evidence, if that enters Ayurveda, assuming, then you will find like this Nadi Shastra, it will be accepted tomorrow. So Ayurveda will gradually change. It may take much longer than in modern medicine, but that change is bound to come historically. So therefore, it's like an evergreen tree, huge, ancient, putting out new blossoms every now and then and remaining young. I'll stop there, and if you have any questions, you are most welcome.
Good afternoon, sir. Thank you for delivering such an enlightening talk. I just wanted to ask a question regarding autoimmune mechanisms, autoimmune diseases. So, have there been successful studies which have claimed to cure autoimmune diseases with Ayurvedic treatment? Have you come across with any of these experiences in your life?
I must admit, I am not an Ayurvedic physician. My knowledge is only theoretical. So about practice like autoimmune disease, can you cure it? What's the long-term result? I cannot answer those questions. Any clinical issue, a patient coming with these symptoms, how do you treat and what is your way of treating? I have no way of treating Ayurveda. Ayurveda, my only interest is I am looking for cues in Ayurveda which can be studied. Like I gave you, that's why one example I gave, Vata Prakriti, a fundamental concept in Ayurveda. Now it could have, I can imagine it having applications in practice. Nobody has done that. For example, if you treatment being done, Vata, Pitta, Kapha, their response is different. That may determine someday our method of practice. If you do this for this type of Vata, you get better results. Your dosage may be much less. The drug that you are giving, instead of giving 25 milligrams, this may require only 5 milligrams. You may find responses because that is based on genomics, as you know. So therefore, that kind of I can imagine having applications. But at the present time, anything about treatment in Ayurveda, I won't be able to answer. Because I was telling this, 30 years, I was only doing cardiac surgery. So I had nothing whatsoever to do with Ayurveda. It is only when I was 65, I decided to study Ayurveda, only for this, for research purposes.
Hello, sir. Based on your extensive collaboration with medical practitioners from different fields, what are your views on the concept of homeopathy?
Homeopathy, I know nothing at all.
Okay, because there are very controversial statements from different regulators, health regulators, but then many people in India, they say that they are getting benefits from homeopathy, so it's always
in
very
fact,
confusing.
that is part of Ayush ministry. As you know, Government of India, officially they have accepted. It is part of Ayush department. But I know absolutely nothing about homeopathy. I know Hahnemann's how it started, because in the 19th century, as you know, The drugs that modern medicine was using, they were actually more toxic than the disease itself. Like arsenic was used, mercury was used, a lot of metals were used. And if you read the European medicine and surgery, a lot of quackery was done. Medicine actually, in modern medicine, the first scientific treatment by giving drugs, it came with penicillin, where we first demonstrated there is a bacterium, the cause of this suppuration, and here is penicillin. 1926, Fleming's great discovery. That's everybody agrees. The first entry of science into modern medical treatment, it came with penicillin. Otherwise, we were not much better off. Lot of quackery. So, but I don't know anything about homeopathy.
Thank you, sir.
Thank you, sir, for inspiring talk. Sir, what's your thought? What's your thoughts regarding entry gut, the using medicated enema treating the nervous disorders in Ayurveda? Opportunities and challenges in the field of entry gut, maybe an enema, medicated enema for the nervous system disorders.
No, you know, the Ayurveda, it is not empiricism. Empiricism is my grandfather, great-grandfather, they all did this treatment. They got good results, therefore I am giving. That is really not science. Ayurveda is not that. Ayurveda, there is a hypothesis in everything, whether it is rasayana, producing good results, or this enema that you talk about. Enema is part of Panchakarma. Panchakarma, there are 5 different acts. One is applying that snehana, you are applying oil, intake of oil also. And then you are svedana, you are putting in a Turkish bath, you are sweating profusely. That is the second. So the body is already, the microenvironment is changed by doing this. And then only you start giving. If the disease affects the upper half of the body, you will give an emetic. If it is the lower half of the body, you will give an enema or a purgative. Or if it is head and neck, you have nasya through the nose. Now these 5 things are there in the Ayurveda. Now I say this because, not because this is my ancestors did this, therefore I am doing. That's not the reason. So Vagpada gives very clearly why they are doing it like this. That's why hypothesis. Hypothesis, without hypothesis there is no good science. I am talking about biology. In this case, Vagpada says, what do you mean by body? Body is a compartment, skin outside and the mucous membrane lining the gut from the mouth to the anus. That mucous membrane, continuous. So skin outside, mucous membrane inside, that compartment, that is body. That is always dark. Outside it may be very bright. And outside pH may be anything. But here the pH is constant. Maybe it varies with outside. Chemical composition, physical, everything is different. There is a disequilibrium between the body within and outside. Only the dead body equilibrates. Dead body, everything is the same. Now this, suppose there is a doshas when they get perturbed, and that I must clarify here, a lot of confusion in Ayurveda is because of confusing this dosha with the dosha prakriti. Dosha prakriti I talked about is a genetic term. But dosha here represents metabolic products. There is a body metabolism going on. This Ayurveda says that. And out of metabolism, 2 products come. One type of product, they get absorbed, assimilated in the body. When we eat food, for example, one part of it is absorbed. It becomes part of our body tissues, dhatus. But there is a part which is refused, rejected. Now the rejected materials, these doshas are part of that. But 3 of them are very important. Even though they are rejected, they are necessary for the body's survival. Vata, Pitta, Kapha. They are different from the other Vata, Pitta, Kapha. Please remember that. Now, these doshas, when they accumulate, body is also this dark compartment which we talked about, is crisscrossed by millions of channels. What is srotas? Ayurveda calls it srotas. Billions of channels, through which all sorts of things are flowing all the time. There must be uninterrupted flow. That is an essential condition of good health. When you say somebody is sick, that means these channels are getting blocked by these metabolic products which are toxic. Now, if there is something blocking the esophagus, for example, or the colon, you can eliminate them easily. You can give an emetic, you can give an enema, you can get rid of it. But if it is in this dark compartment, how do you get rid of it? How do you have access to it? But you have to eliminate. Now, this was the problem which they were trying to solve. And they realized when you eat from this gut, that long channel, things are getting into the nutrients, for example. When we eat food, it is getting into this dark compartment. That's how it's reaching everywhere. So it is porous. It's not a cemented, plastered wall. There is entry into the compartment. So if you give these fatty preparations, oily substances, medicated, they will get into the body compartment through these pores in the gut. And when they get into these srotas, these doshas which are solidified here and there, they will get loosened. Then they are put in the chamber, a steam chamber. They are sweating, all the pores, channels, they all dilate. This is a common experience. When that happens, these loosened dosha, they will come through these openings back into the gut. Then we have access. So if somebody has got a lot of sputum, has got respiratory illness, accumulation of doshas, you give something to make him vomit, all this will go into the stomach eventually. So give him an emetic, he will vomit, throw it out. That is how they eliminate. Similarly, migrating into the lower part of the gut, you give them an enema. So the elimination is done. Now here none of this could be seen. This is all in their imagination. Hypothesis is imagination. But it's creative imagination. Unless you can think, you will not come up with this. And then you find results. Somebody was having all this, lot of, a patient who has got fever, like COVID, for example, respiratory illness, he is bringing out a lot of phlegm. An Ayurvedic physician, this is what he thinks. So when all this is eliminated, he gets better. So Vagbhara says, in fact, about his treatment, idam agama siddhatvad pratyaksha phala darsanat. Agama is the text, authoritative text. What I am saying here, this is derived, mentioned in the text. We get it from there. But that's not the only reason why we use it. Pratyaksha phala darsanat. We see the results. This is what he says. So it is hypothesis based. In fact, I used to get all these research projects. It's very hard in my experience, at least Ayurvedic Biology Task Force. Not too many. We would get about 50, 60 research proposals every year because we used to insist it's only an Ayurvedic group and a medical group. Only a joint proposal we would consider. And we excluded all the phytopharmacology because there are so many agencies giving money for that. So we have very little money. So we insisted. But in this hypothesis based, that's what we want. I would say 10 or 15% there is a hypothesis. Others are simply descriptive. Something collecting information. It may have some use sometime, but it's not exciting. How can you spend your career describing something? You must do something like this, creative thinking, imagination. In those 2000 years ago, if you could imagine all this without seeing anything, no microscope, nothing. So when I read this hypothesis of Panchakarma, that is really good science.
I don't have any subject-specific question, but I wanted to know what inspired you to look into the field of Ayurveda?
This question, your principal asked me this question before I came here. No, that is a very interesting question. I most often asked this question. The reason is this. As I mentioned to you, 30 years I was active cardiac surgery. I was very much involved in developing instrument things which we need to do cardiac surgery. Because valvular heart disease is very common in India. And once the valves they are not functioning, sometimes we have to replace them. And these are imported, so very high cost. In Trivandrum, when I started, we had like 20, 30 valves imported valves we can use. But I had some 200 patients, all young. Average age is 20, 25, and most of them girls. So it was a very extremely difficult situation for us. You cannot tell them who are struggling for breath, your turn will come after 2 years. How can you tell them this? That was the pressing need. That's how we started. And I had only, my colleagues are all trained in India, the engineers. I had 2 engineers, young people, fresh graduates. Never been abroad. These 3-4 people, but we did it. It took 7 years, several failures. And when you fail, everybody will attack you. You are wasting money, government money, you don't know the job, all this, nobody will support you. So we work under adverse circumstances. But there, one of the essential things in India is you have to have the spirit of Bhagiratha. If you fail, you start again. That works. But I changed, I took interest in Ayurveda for a different reason. In 1990s, that is towards the end of my term in Chitra Institute, I found that in the operating room where we do open heart surgery, there will be about 10 people, patient, nurse, doctor, anesthetist, etc. Other than these human beings, everything in that room is important, including the idea of an open heart operation, that you can operate on the heart, inside the heart, stopping the heart and lungs, using a machine to do that job. At the end of the operation, all done under vision, clearly job is done, then you can turn the patient's heart and lungs on. Now this idea, it's not Indian, it's done by an American. All the instruments that we are using, heart-lung machine, ventilators, life-saving equipment, all the drugs we are using, you name it. Even disposable plastic devices, we use it only once and throw it away, that also. Everything is important. Then I realized this is not only in my specialty. Every specialty is the same. Plastic surgery, cancer surgery, you go there, they're all doing the same. This started bothering me, which I was never bothered in my young days. I never thought about it. But the realization of this, the gravity of this, the British brought European medicine here in the 19th century. The first medical college came in 1836 in Calcutta. They established research institutes, hospitals, everything. But in all these years, some 200 years, let us say, there is not a single disease for which an Indian scientist, Indian doctor, found the cause. Bacteria, virus, anything. There is not a single disease. Japanese have. Not only Europeans, the Japanese have. Even Brazil, Chagas disease. Not a single disease. We have plenty of diseases. No Indian found the cause for any of them. There is no treatment, no drug that we developed, which is used all over the world. Not tell me it's used in Karnataka, in Kerala. That's not, I don't accept it. Internationally accepted. There is no drug that we have developed in all these 200 years. Is there an operation that we have designed? Yes, there is one. That was done by Susruta in Buddha's time. Rhinoplasty. Series of operations. He never consulted anybody. He didn't read any book. It was his own. He described surgical instruments. That's an example. That's the only procedure. Otherwise, no surgery, open heart surgery, which I am doing. No Indian developed it. It's from outside. Now, this kind of even theory, conceptual things, there is no such thing that we have. On, say, for example, tissue immunity, the exciting days of transplantation. There is a whole lot of highly imaginative theory about self, not self recognition. If you put any other tissue, even identical twin, it will not be accepted. After some time, it will be rejected. How did this come? Very interesting theories about all this. No Indian has come up with anything. So this started bothering me. Are we incapable of innovative thinking? Then there was a great, I should not call him great, very important physicist called Shockley, who got Nobel Prize in 20th century. Shockley said once I was in the United States at the time, he made a statement that original thinking, groundbreaking, new ground being opened, that is only white people are capable of doing that. He said this. Others may make contributions, something useful, but something revolutionary that only white people are capable of doing. Everybody abused him, criticized him, but he stood his ground. He refused to change. I started thinking maybe Shockley is right, or partly right. I was very upset. I was not willing to admit. But then I thought, after all, this medicine came here only 200 years ago. We have been treating people for thousands of years. We had Vaidyas everywhere. A lot of people go to them. How did they treat? They have written texts. Why not look at them? That is how I first started looking at the Susrutas, Susruta Samhita. It was an English translation at that time. Then I realized, all these, operations, these are all new things, which at that time did not exist in London or anywhere, because it was Susruta's rhinoplasty seen by 2 British doctors in Pune. All this I have written also on that. That was published in a magazine called Gentleman's Magazine in the 18th century in England. And a British surgeon saw this, and he did this, the first rhinoplasty. Now, that was original. So I took heart in that. That is how I got into Ayurveda.
Thank you, sir. Thank you, audience. May I now request our principal and vice principal to escort our chief guest for the felicitation program. On behalf of everybody present here, I would like to extend my heartfelt gratitude to our chief guest, Dr. Valiathan, for taking time from his busy schedule to be with us here today and for inspiring us with his kind words. As we've reached the end of this program, I would like to propose the vote of thanks. My sincere gratitude to all Mahe officials, dignitaries, our principal, vice principal, faculty, research scholars and students for your participation and for being such a wonderful audience. Once again, thank you one and all.